Hello and welcome to a new chapter called as Gene, its nature, expression and regulation. In this chapter, I'll be introducing you to the fantastic molecule called as DNA, and we'll be also looking at the central dogma of protein synthesis. So let's begin with this module and this chapter. Now in the previous chapter, we have studied that offsprings inherit their genes from their parents, and this concept is called as inheritance. This pattern of inheritance was explained by Gregor Johann Mendel, and you saw that in the previous chapter. Okay? Gregor Johann Mendel was also called as the father of genetics. So what exactly is this molecule called as gene? Now gene is nothing but a segment of DNA which is responsible for the inheritance as well as expression of that character. The term gene was coined by none other than Johansen. Correct? Okay, moving on. Now what you see here in the slide is a family of rhinoceros. Next to it is a family of human beings. Right below is a family of zebra, and next to it is a family of the penguins. Now in all these four pictures, you basically see living organisms of different types with two generations each. Okay, means the parents and the offspring. Now living cells and living organisms have this remarkable ability to reproduce and transmit characters from generation 1, which is the parents, to generation 2, which are the offsprings, correct? And this is what we call as inheritance, right? Why do they do that? In order to maintain the continuity of inherited traits. Yes? In the 20th century, one of the landmark discoveries in biology was the identification of DNA as the genetic material of living organisms. What is DNA? This is the molecule of DNA, and DNA is nothing but deoxyribonucleic acid. You have to remember this full form of DNA, deoxyribonucleic acid. Moving on. Now, let us look at this diagram right here. You see a structure right here. Now, can you tell me what structure is this? Definitely, this structure is that of an eukaryotic cell. Now, why it is called eukaryotic cell? What does the eukaryotic cell contain? A eukaryotic cell contains a nucleus, and that is why it is called as eukaryotic. Now, what does the nucleus has? The nucleus basically has a structure called as chromosome. And what is this chromosome made up of? The chromosome is nothing but DNA molecule wrapped several times. So chromosome is made up of nothing but DNA. We'll be studying in this chapter the inheritance and the expression of DNA molecule, and that is why the chapter can also be called as molecular basis of inheritance. Right? Moving on. Now, DNA is the genetic material of living organisms. What are the criteria that must be met by a genetic material to be called as genetic material? It should have the following criteria. Number 1, it should be chemically stable. Under no condition it could degenerate or it should degenerate. It should be chemically stable. The next criteria it should meet should be it should express accurately. Right? DNA basically is made up of genes, and genes express themselves, correct? The next criteria is it should be able to replicate itself with minimum errors, and DNA has that ability to replicate itself and make its own copy. And last but not least, it should also have scope for mutation, which is scope for change, and then this change is transmitted to the next generation. Now, since DNA meets or passes all these criteria, DNA is the genetic material. Okay? Once again, for someone to be a genetic material, it should have the following 4 criteria, namely chemically stable, express itself accurately, replicate with minimum errors, as well as have a scope of mutation, and all these criteria are met by DNA. Moving on. Now in this chapter, we'll also see that, or we'll also define gene as a segment of DNA having information for the synthesis of a particular protein. Now, how do you think that gene or DNA decide a protein? Very simple. For that, we'll look at the next slide. Now, DNA, we know that is deoxyribonucleic acid, undergoes a process called as transcription to produce yet another nucleic acid, which is called as RNA, which stands for ribonucleic acid. Now RNA then goes through a process called as translation and is translated into a protein. Does that make sense? What you look here is a protein. It is nothing but long chain of amino acid. Every circle there represents an amino acid. So, putting it again, DNA undergoes transcription, makes RNA, RNA undergoes translation and makes protein. Now, in a eukaryotic cell, the process of transcription will take place within the nucleus, while the process of translation will take place in the cytoplasm. And this entire concept right here is also called as the central dogma of protein synthesis. Now, this idea was put forward by Francis Crick in the year 1958. Is that okay? Now, what is dogma? Dogma is nothing but a belief that is considered to be absolutely true. So we have to believe and we have to look at this picture and remember it for the rest of the chapter, because this is the gist of the entire chapter. That DNA molecule undergoes transcription to make RNA, and RNA undergoes translation to make a protein, and this is the central dogma of protein synthesis. Once again, so what is the central dogma of protein synthesis? Crick, Francis Crick, he proposed that DNA determines the sequence of amino acid in a polypeptide or a protein through messenger RNA or mRNA. Is that okay? Now, this brings us to the conclusion of this module, where we introduced what DNA was, correct? And we also talked about the central dogma of protein synthesis. In the next module, we'll be looking at certain experiments that had been done to prove DNA as a genetic material, as well as we're looking at Griffith's transformation experiment. Okay? Thank you and take care.